The first talk of the second section is by Adrienne Fairhall. Adrienne is a professor at University of Washington in Seattle. She is well known for a variety of work in theoretical neuroscience, and I gather now famous for an online course in computational neuroscience, which all sorts of unexpected people have taken. Anyway, Adrienne's going to talk about rules of adaptation across Cortex. So, welcome.
Thanks for the introduction, and thanks so much for the opportunity to be here. It's really amazing meeting, learning a lot. So I'm going to give a little bit of an outlier talk. Given the theme of canonical computation, I did want to concentrate on a recent project that we've been working on, and to remind you that one of the most canonical computations in the brain is adaptation. It happens everywhere, it's completely ubiquitous, and I think I'm sitting here and my talk is at a great point between Eero's talk, where he raised the issue that there's lots of dynamics and biophysics that AI models are currently not taking into account, and Nicole's, where she's going to be talking about adaptation at a somewhat higher level than you'll hear about from me. So I did want to maybe preface with just sort of warning you that this is old school stuff I'm going to be talking about, but I'd like to kind of frame that as, you know, so old school that it's kind of steampunk. So try and see it in that frame. All right. So what is adaptation and what's it good for? What the key issue is that neural responses are fundamentally dynamic. When we think about the neural code, we really do have to think about it as something that's continuously changing. Neural responses depend on stimulus history and, or maybe via, their own history of activation. And why do we think this happens? You know, ideas have been out there for a long time that this reduces redundancy, increases efficiency of coding, and it allows neurons to track time-varying input statistics and adjust their response accordingly. And so just to make sure that I don't disappoint Tony in putting up a slide he's seen before. So this one, this is just an example of where, you don't remember this one? Okay, great. So this is an example of where we see that. So for example, in single cortical neurons, if you drive them with stimuli that have different variances and you extract their input-output curves, you get a family of different input-output curves. When we normalize those input-output curves by the standard deviation of the stimulus, they're all the same. And so individual neurons across different sensory areas have the ability to encode stimuli in units of their time-varying variance. That is only one aspect of adaptation, many others, and in fact, there's a whole family of ideas and phenomena out there that seem like they should be related, but at the moment we don't really have a theory to pull them all together. Things like oddball paradigms, when you change the probability of two different stimuli, neurons adapt their responses such that the more uncommon stimulus has a larger response. That is thought to depend on issues like repetition suppression. We have also stimulus-specific adaptation, where things adapt to one particular stimulus. The concept of mismatch negativity is out there. How does that relate to all these other things? All of these ideas sort of have under the hood that they seem to imply that neurons or neural systems are able to do some kind of online statistical learning, and so give us kind of the primitives of the ability for even very low-level neurosystems to do a kind of inference and potentially underlie things like predictive coding. So all of those questions sort of inspired us to set up a collaboration. And so where I want to go today is to talk sort of generally about how is information, temporal information in particular, processed by Cortex. So what you've heard a lot about is how visual information, you know, frames of images that are flashed up, get processed, and we starting to understand a lot about the mechanisms by which that happens well. The real world is time varying, things are changing all the time. Neural dynamics is complex. How do those things kind of interplay? We'd like to understand how the rules of information processing potentially differ across cortical areas. You know, one of the fundamental ideas of efficient coding is that the responses of neural systems are matched to the statistics of the input. Very good reasons to think that those statistics differ for different types of sensory stimuli. So do we see any evidence or substrate for that in the processing properties of different parts of cortex? Are there differences in these rules among cell types? I think cell types are very important. They have different biophysics. You know, do we see any evidence for differences in processing rules at the level of cell types? And finally, we'd like to understand, so what might these kinds of short-term dynamics that are sort of under the general term of adaptation, contribute to coding and memory. And I think this is where I'll probably hand off the bigger picture thinking on that to Nicole. So in the service of this goal, we assembled a team with Human Frontiers funding to look at these questions across different parts of Cortex. So Ilan, Nicholas, and Ellie and I formed a collaboration through Human Frontiers. We gave ourselves a logo inspired by this one, but as you can see, we're a little more modest because we only have Human Frontiers funding and not Wellcome Trust and Simon's funding. But the key player really in all of this project is Kenneth Latimer, who's a postdoc with me that I inherited from Jonathan Pillow, who was a child of Arrow. So it's kind of embarrassing at this point. We're actually looking at scientific grandchildren, which we still think ourselves too young to have. All right, so common rules of information processing in Cortex. We had big visions of how we were going to do this. Would have liked to give noise stimuli with interesting time-varying envelopes, as we've been able to do in single neural systems, but that's not really practical when you're trying to design the same experiment that you can run across different cortical types. And I think we're hitting the same kinds of problems as the internet, or not problems, but design issues as the International Brain Laboratory is. Or what kind of stimuli and experiments can you do that are doable in multiple labs? And so what we ended up converging on is a very simple kind of stimulus, very classical, where we gave each of these different neural systems sort of pulsatile stimuli, right? Because there we can really design things that we think are sort of comparable from one area to the other. So in V1, we just gave full field flicker. In S1, hits of whisker deflection. And in A1, we gave clicks. In the 3 labs, they used the same mouse strain, same anesthesia. So again, steampunk, we're working with anesthetized animals. And we're doing, again, steampunk, whole cell current clamp recording. So these are actually intracellular. So I know probably, except for Eve, maybe the only intracellular recordings of the entire meeting. All right, so how do things look? So let's start with some very simple cases. So we gave these sequences, 1 Hertz stimuli, and you get these responses from V1, increase the frequency of the stimulus, and as expected, what you find is that these pulses are eventually smoothed over, so you're not able to resolve individual pulses. As everyone knows who works on mouse, these are incredibly variable, and so there is actually tracking in some of these individual cases, but it kind of washes out in the average case. So that's what we get in V1. Here's what it looks like in S1. Again, you get very nice tracking to the 1 Hertz that kind of disappears as you increase the frequency. And I'll come back to some of the details of these a bit later. Here's A1, again, tracking, maybe see it a little bit nicer all the way through these different frequency ranges. So some of the things that you're seeing quickly at a glance is that there seem to be some timing differences in the ability of different cortical areas to track stimuli. Where we hope to go, at least with understanding this population data, is maybe to be able to account for things like these experiments from Tony Zader. So Tony, a few years ago, took a related approach in looking at the behavioral consequences of timing in different parts of cortex by stimulating V1, A1, and S1 with pulses of inputs and seeing whether animals could be trained to discriminate. And so hopefully what we would be able to extract are the reasons for these differences in behavioral timing. But what I hope you noticed in that data was that the responses to 1 Hertz are actually quite surprising, at least were surprising to me. So as a response to each one of these pulses, you don't see a nice clean response. You see this very extended waveform in the voltage that has this double peak. That's true also in somatosensory cortex. When you stimulate a little bit faster, you don't see that anymore. But look at the time scale of this. This is a response that's going on for 500 milliseconds, so it's really very long. We see that over and over again. You get this very complex single pulse response, both in V1 and S1, and I'll show you later why I'm not really focusing anymore on A1. So we also saw, some of you would have noticed, that at the end of this long train of 10 Hertz pulses, you very frequently get this off response. So there's a large kind of rebound response at the end of the sequence. If you stop the sequence in the middle and you unexpectedly add in a gap of sufficient length, that off response turns into what looks like a kind of emitted stimulus response, like as though there's a kind of surprise that the sequence stops and continues again, and you see this off response at the end. All right, so we see a variety of relatively complex behavior, even to these very simple stimuli. So us being us, what we'd like to do is try to capture that with some kind of model. And since I've inherited Kenneth, I've also inherited Jonathan Pillow's slides, so I'm going to use Jonathan's images from now on. And so here, what we're trying to do is to fit the standard kind of generalized linear model, see if we can capture these long timescale complex responses through these kind of cascade models. So we're going to fit a filter, we're going to assume a nonlinearity, include a post-spike filter, and see how well we capture those responses. So obviously we can't fit them from our periodic data. We need to stimulate with richer stimuli, and so in the case of pulsatile stimuli, that means we're going to drive it with spikes of input, but that are distributed in a Poisson way, so they occur at random times. And so here's an example of what we get from fitting such models to V1. Why are there multiple overlaid? That's because we fit it with Poisson inputs with different frequencies. And as we all know, if you try to fit such models with different stimulus statistics, you get different models, and that's very apparent here. Same in S1, we get actually quite similar family of filters that we get by fitting at these different frequencies. And so it's not very satisfying. We'd like to do better. So one solution to that is to, rather than fit with a particular rate of Poisson inputs, we now fit with many rates, where we're going to take different blocks at different rates, add them all into our stimulus set, and try and fit some grand model to all of those different stimuli. We're also going to use an idea that Kenneth has been working on in collaboration with Jonathan. And here, this is an idea that they first applied in some work previously with Frederici, looking at responses in the retina. And so in the retina, what they were particularly interested in is trying to be able to capture independently excitatory and inhibitory inputs. And to do that, they split this feed-forward model into different filters that pass through independent nonlinearities. And that is nice because that now allows you to find a family of filters that can each be treated independently. And it's similar to work we've done in the past, where we use covariance methods to split out different filters. We're taking a kind of GLM version of that idea, where now these different filters we can think of as sort of representing subunits of input that are passed through independent filters. And so obviously we've added a lot of parameters to that model. That's going to give us a lot better chance of fitting the data, and indeed it does quite well. So we fit using these Poisson trains, and now here's the response to a Poisson sequence. We can get quite a good prediction for the responses of the voltage to those inputs. And what you'll see is emerging here in the filters is you get some that incorporate these really long time scales. So just look at the time base here. This is out to 2 seconds. So we have influences from inputs that are going on for multiple seconds that we've been able to expose by using these sort of long time constant inputs. So how well does that do in predicting some of these inputs? So now, because we have multiple filters, we're able to capture these complex responses to single pulses and to the 4 Hertz, and then we see the loss of tracking of the high frequency inputs and these offset responses. And those different influences kind of split out into different subunits. So this biphasic pulse response is accounted for by one subunit. The offset response is taken up by a different subunit, which is actually not what we expected to happen. So it's kind of good that those show up in different subunits, because this example in S1 shows you that you can have a simple, non-biphasic response to a single pulse and get the offset response. So clearly they don't depend on one another. This offset subunit captures that emitted stimulus response. So you see here that orange subunit that captures the off response is also capturing that response of the system when we pause the pulse train. So just to quickly flash up an A1 response, in general, at least in Ellie's hands, the A1 responses have been just much more variable, and so we just haven't been as confident in the model fits. Just wanted to show you one example. We're doing okay, but it's kind of a mess, so these are individual trials. So is there a role for different cell types? So many of you will be familiar with Maria's very nice recent paper, where she was able to identify, in the case of stimulus-specific adaptation, how PV cells and somatostatin cells seem to be playing slightly different roles in governing the gain control in stimulus-specific adaptation. So we divided out our neurons by cell type and had a look at that. And so maybe this is even the first cell type slide that you've seen in this entire meeting. So if we look at PV cells, hopefully what you'll see is that that is actually very similar to what you saw from the excitatory neurons that I showed you before. They show all the same responses, this kind of biphasic response to single pulses and gradual tracking disappearance of the time locking, and also that off response at the end. So that's PV cells. Here's somatostatin cells, again, very similar, sort of statistically indistinguishable as a population so far, at least at the amount of data that we have from the excitatory cells. So they all seem to have pretty similar behavior. Some of you will remember this very nice paper from Michael Berry, where Michael showed that some of these things are already there in the retina. So this is work with Greg Schwartz. So here they were giving sequences of step inputs and saw what they called a time and emitted stimulus potential, harkening back to work by Bullock in the olden days. And so you see this response that occurs at the end of a sequence of pulses, and they did a bunch of nice work characterizing that. So we're not surprised to see such responses in cortex, given that they're there very vividly in retina. So let's have a look at what's going on in the middle in LGN. So in LGN, these are all now extracellular recordings. We don't have so many of them, but LGN already has these complex responses to pulses, and a lot of the effects that we see in cortex are in fact already there at the level of LGN. So what happens if we look in LGN where we block, where we silence cortex? So here we have, or Nicholas has channel rhodopsin expressed in PV cells in cortex, shown on the laser. You turn off cortex, and now what you see is that there are these changes in that rebound, or the second part of that complex response, is actually diminished. And so that gives us some evidence that this part of this complex response is actually due to thalamocortical reverberation. So Michael also showed in this emitted stimulus potential work that there was a time delay between the end of the sequence and the arrival time of the emitted stimulus potential that actually scaled with the frequency of the pulses, which seemed to suggest that the system kind of knew the frequency at which it was being stimulated, and it got a rebound response that was proportional to the driving frequency, as you can see in this plot. So is that true here as well? Somewhat. It's a little bit harder to see because remember we're measuring voltage. And so this is one example that's very noisy. As one changes the frequency of the sequence of pulses and you look at the rebound response, you do see a systematic shift in the latency of those rebound responses. So this is one example. We have many. They're all equally noisy, but they generally do show this systematic shift. Does that come out of the model as well? A little bit less clearly. So again, remember the model is modeling membrane potential. So what we get is, in this case, we see that the peak seems to happen at about the same time across different frequencies. But if you were to imagine that the membrane potential is driving a spike and the spike happened at some threshold, then that would give a systematic change in the latency of the spike that's being produced. All right, so just to summarize what I've told you. So here, what we see is we get complex multiple timescale dynamics from these simple inputs across cortex, which can serve as Michael argued in the retina, kind of substrate for temporal pattern detection. These responses can all be captured by relatively simple filter models. In this case, incorporating several filters allowed us to address dynamics that span multiple timescales. We do see timescale and some feature differences between different cortical areas. Some of these responses, in fact, maybe the majority, may be inherited from cortex, although some aspects of the response seem to be thalamocortical. And at this point, we don't see much differentiation in response by cell type. And so presumably the kinds of effects that Nicole pointed out, cell type dependent, are more about the way those neurons are wired together than they are necessarily about the inputs and the responses that they are providing independently. So of course a big caveat that all of this was done with anesthetized animals. Painfully, the noise level and the variability in the anesthetized animal is already very large, so it's unclear, whether, how this is all going to pan out in an awake animal. All right, so what does all this say? So what this tells us is that some of these aspects of adaptation should really be better thought about as just long timescale properties of the stimulus. And this is an argument that we've made before. We see complex long timescale dynamics. People have seen, for many years that you start to show a stimulus, you see the response decaying away, the cell is kind of getting tired, right? It's just failing to respond. That's not at all what's happening. In fact, what's happening is that all of those responses are part of the encoding of the input, and they are well described by features of the input that extend over many seconds. And this is something that we have pointed out before in previous work. We showed some time ago that current injection into single cortical neurons generates responses that, at least for this set of neurons that we were looking at, look like fractional differentiation. You get power law-like filters on the envelope of noise. So in this experiment, what we did was to drive the neurons with a noisy input, and we varied the envelope over time. And you see, so what we might have thought about as adaptation to that stimulus envelope, but what we showed is that the form of that adaptation was actually just a transformation of that stimulus envelope, and one that had many timescales of response in it, and behaved very well as a linear filter of that envelope, albeit one that contains multiple timescales like a power law. And we were able to predict beautifully the responses of neurons to complicated sequences of changes in the variants just by using this simple model. And so that led us to argue at the time that what you're seeing in these sort of slow forms of adaptation to stimulus envelope is a combination both of very precisely timed responses to the details in the fluctuation in the input, but this overall modulation, slow changes in firing rate that are driven by these slow changes in the envelope. And that's very consistent with what I've just shown you, is that we can predict the responses of neurons to these sequences of pulses where you see time locking to the single pulses, but you also see properties of the responses that are driven by the overall envelope and the time since the previous stimulus. And so more generally, I think there's an issue that we don't talk about much, which is that the intrinsic properties and the properties of small circuits allow us ways to sort of maintain information about stimulus history in neural excitability, right? So we see here, at least over the timescales of seconds, that there's long timescale maintenance of history in neuronal excitability. Very nice review recently by Nicolas Brunat and collaborators to point out that neural excitability is a mechanism for memory that we often neglect. And certainly in training artificial intelligence models, it's all about the synapses, where there's a lot actually going on in long timescale changes. And I think Eve really beautifully made this point in her talk the other day. And so we've been playing around with some models of that, not that the fact that neurons can have, say, power law-like dependence on their inputs means that they convey, that they maintain information in excitability over long periods of time. We've been playing more with the idea that they may become hyper-excitable, and there are some ideas about the biophysics that would lead to that. And so what does that do for you? If you have some neural network where you stimulus drives some sequence through the network, and these neurons maintain some memory in their excitability of the previous occurrence of that stimulus, the next time you drive it, it's much easier to fire those neurons, and they can potentially even fire one another sort of accidentally just through noise. And so such a model easily produces replay. So if you drive a sequence through the network and then you just trigger one of those neurons, because that sequence is now somewhat more active, it's quite straightforward to get replay events purely through changes in single neuron excitability. And so you can see, you get many types of pathways through the network. And you also capture things that are seen in data from people like Valentin Dragoi and others of spontaneous replay of previously observed sequences that occur in V1 and V4. All right, so just want to underscore that what we see is that single neurons, synapses, and small circuits have a tremendous amount of dynamical richness, which must influence the way that they process and that we are able to understand ongoing temporal stimuli. I think Arrow's idea of sort of trying to understand the way temporal sequences of stimuli, how they move through stimulus space, all of those issues have to be understood in the light of the fact that neural systems have their own intrinsic dynamics that are evolving on timescales that are comparable to the timescales of change in the stimulus. So these dynamics have the potential to add computational richness to biological networks. In the case of adaptation, we think that they're doing lots of smart work for us in dealing with sort of local changes in stimulus statistics. But I think that there's a lot of room for AI models to start to incorporate these types of dynamics, and it'll be interesting to see in the long run where that goes. All right, so that's my acknowledgements. Again, just flesh up our little group, and I just particularly need to highlight the students who took the data, so Michael Sokolexi and Dylan in Nick's lab. All right, that's it.
Thank you. So we have time for questions, and Joshua.
Hi, thanks for the great talk. Regarding the experiments with the hyperexcitability in the sequence that was remembered, is there something in the experiments that let us differentiate the hypothesis that this were changes in the synapses rather than in the soma?
Those were experiments, so that's how. This is all modeling. So we've been playing with different biophysical mechanisms that have been introduced in the literature for how that hyperexcitability might come about, but we've really just been playing with models to see how easy it is to get that to work and how many sequences you might be able to store through hyperexcitability. What are the constraints on whether that might explain the data that's out there?
Okay, so it's a proof of concept.
It's a proof of concept.
But there's other biological evidence that there's these adaptations more in the soma than in the synapses?
Yes, but it's not yet published, so I can't share that with you. So, yeah, there's actually some very nice data coming out, I think, soon that some of these ensembles that get burned into groups of cortical neurons, what they're seeing the change to be, if you look intracellularly, is in the excitability of the neurons rather than in the synaptic potentials.
So if I can actually take the prerogative of the chair and follow up on that, I mean, I think at least in visual neurons in visual conditions that I know of, there are definitely both cellular and synaptic mechanisms, because there are some mechanisms, for example, Matteo Carandini described when he was the firster, which are cell-wide mechanisms. David McCormick has also looked at these potassium conductances that persist. But there are also mechanisms that affect some inputs and not others, and they seem almost certain to be synaptic, right? So, I mean, the answer is, as usual with adaptation, yes and yes again. So, Esther.
Yeah, the question is about the receptor mechanisms and how much of that is, I'm thinking in the somatosensory system about something like piezo2, which is a huge molecule, and getting it to move into an open or closed state might take time, and that might be.
Right, yeah, absolutely. So what I showed you is that at least in the visual system, at least some of these issues can be tracked back to retina, through LGN. I think that must be true also in the somatosensory system, and we know that all the input types of somatosensory neurons have adaptive properties of their own, which presumably contribute to this. I think it's still interesting to be able to look at the level of cortex. And if it turns out that all of those mechanisms in 3 different areas are sort of conspiring to give you the same kinds of dynamics in cortex, that's already interesting. So presumably some of the details in time scale, many of them must come back to the precision of the receptors at the front end. But if cortical circuitry is very similar across different parts of the brain, presumably that does that either maintain or filter out the differences in timing that we know are able to be differentiated at the front end. So I think it's still relevant to look at that representation at this level.
Really nice talk, thank you. I wanted to ask maybe a tangential question, which is the fact that you see more variability in A1. Do you have insights on that specifically? Do you think it's in the stimulus, something about how you present the stimulus, or do you think it reflects a difference in the circuit?
Yeah, I wish Nicholas were here to answer that, because he spent some time in Ellie's lab to help sort of set up the paradigms. As not the experimentalist, I would kind of hate to speculate on that. I'm not quite sure how uniformly we can really think about the experiments that are being set up in different places, or if the animals, we hope that they're under the same stimulus conditions, but we can't really know. That might be real, or it might be a experimental setup issue.
Anne.
That was a great talk, Adrienne, and your logo is really beautiful. I like it. I had a technical question about your thalamic manipulation and the frequency dependence. So you saw a big change in the temporal dynamics at 1 hertz, not at the higher hertz. Was that also present at the lower frequencies as well? Was it maybe attenuated at half a hertz?
No, so there was one figure that had an even slower stimulus where it was every 2 seconds, and you see the same kind of complex dynamics in response to those as well.
Okay, so as long as it's below a particular frequency, then those changes are always apparent.
Right.
And that was only in V1, those experiments, presumably, right?
And S1.
Okay, thanks.
Yeah, we haven't seen such a strong effect of the bimodality in the responses that we've been able to get from A1. So it seems more.
Matteo in the corner.
All the way here. Hi. You showed the first page of a beautiful paper by Tony Zader, which is a timeless paper because he did that with electrical stimulation when anybody else would have used optogenetics, and he found great results. Steampunk as well. And I was wondering, unless I missed it, whether you can go back to it and predict those results. Maybe it's worth summarizing them because not everybody might know that it's a little gem of a paper.
Yeah, that's right. So what Tony did was to give sequences of inputs that are separated by a particular timing and train animals to see whether they could detect differences. So it was the idea to see to what extent timing mattered in different parts of cortex. And so then you plot percent correct as a function of the interval between the stimuli. And what he saw, if you recall, was that different parts of cortex, when you stimulate different parts of cortex, you see response curves where V1 was slow relative to A1, which was slow relative to S1. So we would love to predict those. That paper just sprang to my mind a couple of days ago, so we don't have quite yet the data to go back, but that's definitely in the plan. So I was happy to remember it in time for this talk.
On the aisle.
So you touched on the relationship between adaptation and efficient coding and reward statistics. I was wondering, do you get the sense that the filters that you identify can themselves be flexibly adapted to the environment, or are they hardwired into the biophysics?
Yeah, that is a great question. So in other examples that we've looked at, and Tony and everybody, right, when you change the statistics of the stimulus and you extract a filter, you will get a different filter in general. Where we've tried to avoid that here is by stimulating with this variety of different firing rates. So hopefully what we're finding is a model that's kind of maybe the grand ensemble over those different models that we would have gotten if we'd stuck to a more restricted state. And so I think it's a really interesting and key question as to what extent does the receptor field that you ever extract from a particular experiment really correspond to a true receptor field, or if it's just a particular sampling that's right for, or that is produced by the stimulus that you're using. And in this case, we've hopefully avoided that. But that's not to say that if we now came back and drove the neurons with, say, noise or some other kind of stimulus, that these would still be good receptive fields to predict the responses to those. So Steve Backus has a very nice paper with, I think with Surya, where they've been using machine learning to try and, sort of deep networks to try and predict the outcome, so you don't, where you throw in sort of every kind of stimulus you can imagine, in that case into the retina, you get very nice predictions where you don't sort of ever really look in too much detail of what the receptor field actually looks like. So again, this is sort of, what are we trying to understand, right? Are we just trying to predict outputs? Are we trying to get some sense of what the effective computation in a way that we can sort of understand it in some low-dimensional space really is?
Terry.
So in the case of clicks in the auditory system, that's very broad frequency, right? And when you record from different neurons with different preferred frequencies, do you see any differences in their filter characteristics?
Yeah, I think hopefully the subtext of part of this talk is we don't have as much data from A1 as we would like to have, so I can't answer that question. I think Ellie's not been quite as committed to this project as the rest of us, so hopefully we'll be able to say something about that in the long run.
He looks very committed on that picture.
In this slide, yeah.
Very nice talk. So I was wondering, so you choose to record parallel on the primary sensory cortex, but if you think about it, and you find that A1 was a little bit different, and in Tony's lab, we always thought that A1, if we counted the synapses, that it was from the periphery, and it's really much farther away. So I was wondering if you, instead of doing A1, you have done something like inferior colliculus, then it will be a much
more
homogeneous compared to the S1 and B1.
That's very possible, absolutely. Yeah, that would be a great thing to do too. So, in an ideal world, Nicholas has gone on to look at LGN. It would be great if Ilan had time to go and look at POM and VPN. It'd be great if Ellie was able to. But as I mentioned, our funding sources, Human Frontiers, gives us, I don't know, $85,000 a year or something. So we're limited in what we can do at the scale of this project. It would be great if we can continue it at a more advanced level.
Okay, if there aren't any more questions, let's thank Adrienne.